Hi guys, welcome to class. We'll get started maybe like right now. Yeah. Okay. Okay. So we're doing week 6 like this, right? So it's preference. If you want a paper copy, you can ask office for it. So next week will be week 7. We're doing free response again. So we'll be alternating. So this way you get a feel of what the sample questions are. Kind of get used to it. Yeah. So, and the other thing is that try to submit the test. If it's multiple choice, you can submit them online. So if you look at the, I think the assignments, like test number 6. If you look at this one, so if you look at the instruction here, you have the Google form. Yeah, Google form. So you can click on the Google form and key in your answers. They'll score it for you. Yeah. So that way you know which question you missed. I actually think it's very useful when you go through these tests. You should circle the question that is difficult so that you can ask them. So sometimes you guess question correctly, but we're not sure why it was done correctly. So by making a record of it, it's a little bit better. Okay. So as a reminder, always, if you can turn on your camera, that would be great. Yeah, I can't really enforce you guys to do so, but if you can turn on camera, that would be awesome. Okay, guys. All right. Let's take a look at the test. Yeah. So I'm still going to put the test online in case. Open the test here in case you don't have a copy of it. Let me just download the test. A little bit easier. Okay. And multiple choice test, you're going to see maybe 7 or 8 data in a set kind of question. Yeah. So then they'll give you a bunch of data in the form of table, figures. Those are usually experiments. And there's like 4 or 5 questions follows. Those are usually the hardest. So if you want to save time on easier questions, the strategy for multiple choice is do as fast as you can for easier questions. Yeah, okay. Do as fast as you can for easier multiple choice, and then save time for data in a set question. Is this clear? I'm not sure. Is that no? Because I ran out of my contacts. I'm using old pair, so everything I see is a blur. I can't tell if it's clear. Okay, so this is the multiple choice. Alrighty. So, all right, let's get started. So if you can turn on your camera again, yeah. Just sometimes. Yes, it's better if you can see your face. Yeah. Okay. Right. And how do you guys feel about school? There's no more math requirements at school. How do you guys see a lot of people? You feel more comfortable with masks still a little bit? Yeah. What? It kind of takes a while. Every time I go outside, I still put a mask on. I have to talk, so I have to leave the mask off. But when I go to the gym, I see about a third of the people with masks. Most of the people just take it off. So, but anyways, I just was wondering how you guys feel about that. Okay, so let's take a look at this. I'm thinking one way we can do this, I might as well just go through the test quickly. So some of the questions that you would have, or some of the questions you know already, maybe the answers to. I just wanted to kind of go over, make sure that we didn't miss anything. Some questions are good for analysis, and this is the kind of question. Now you'll see there's not a lot about memorization. Okay, let's see what this looks like. So number 1. Okay, so again, I'm going to try to use both our notes. Maybe you can take some notes at the same time, answering some of the questions. Okay, like that. Okay, it's a little bit hard. Again, I only have one screen here. At home, I have 2 screens. Hey, okay, maybe we should, yeah. Okay, so let's look at question number 1. They're looking for hypothesis that a large object collided with Earth, meteor, right? In a period of time associated with mass extinction. The last one was 65 million years ago. It was a catastrophic event that wiped out the dinosaurs. So the question is asking to find out about meteor. The evidence of meteor is deep. So if you didn't know this, iridium are actually an element found in meteorites. And on the sedimentary rocks, the layer where dinosaurs were, there's a high level of iridium, indicating a meteor impact. So the organisms that were around in the same time were exposed to iridium. So that was the evidence that we know. And then we were looking for the crater for the longest time. Finally, we found the crater in an undersea drilling company, found the crater in the ocean. So that kind of validates the hypothesis, okay? So that's easy. Number 2, which one represents a mutualistic relationship when both are benefiting? Now, if you look at number A is not a mutualism. If you eat others, this is predation, yeah? Okay. B, if you look at algae, provide sugar for use in cell respiration. This is not a mutualism because you're not involving another organism, right? So the answer is D. So flowers has been mutualistic relationship with insects, with birds, and with mammals for the history of Earth, yeah, since flowering plant evolved. So I want you to know the term for flowering plant a little bit. So let me this. The term for flowering plant, you should know they are called yeah. Now the most primitive plant, bryophyte, is the most primitive plant. Bryophyte, barely mated plant. So bryophyte, and then we have sperms, and then we have the gymnosperm, and then you have the flowering angiosperm. Okay, so those you can think of them are the 4 phylums or divisions in the plant kingdom. Okay, so just know a little bit of those. Okay, we'll come back to them if we see more detailed questions about plants. Number 3, we have a blue flower. So blue flower African violet of known ancestry self-pollinate. Now, a lot of people saw this word self-pollinated and right away call plants, flowers, asexual. Now, flowers, whether they cross-pollinate or self-pollinate, they go through meiosis. They are sexually reproducing organism, period. Yeah. Self-pollination does not mean you're asexual. It just means that the meiosis product, the sperm cells carried by pollen, and the meiosis product, the eggs in the ovule, yeah, they fertilize themselves. Okay, all right. Usually those flowers are more drape, wind pollinated instead of attracting insects with nectars or scent or color. Yeah. Okay. So here you have 36 blue, 14 produce pink. What is the best explanation for the pink? Well, the answer key says B, right? Pink flower is recessive. And how do we know this is recessive trait? So this is about 3 to 1. Yeah, the best we can. They don't give you exact number. You know, exact number. You know, in reality, nothing is really exact, but this is the closest 3 to 1 ratio. And you only have 2 phenotypes, so you're not going to be thinking about what is that incomplete. Because incomplete, then you're going to get 3 different colors, right? So that's pretty easy. So the answer is B. Number 4, okay, while there are only, this plant, again, this is the phylum of the plant. Let me just put something that I forgot to do this before. So I'm just going to put a note here in case we see this phylogeny. So if you look at the plant evolution, the first, let me draw a little bit longer line. So it is not something you have to memorize. I'm sure this will be given, but this is just something you should know, that the first plant that branched out are the bryophytes, the most basic plants. These plants are moss, for example, okay? They form the carpets of the rainforest or forest. They don't have anything, they're barely mated plant. So these ones have no vascular tissue because vascular tissues like xylem and phloem, that make up root, stem, and leaves, evolve later. So vascular tissues, we do need to know, we'll see the term xylem, which transports H2O, water, right? And phloem sounds like food. So this is what transports the sugar. So those vascular tissues, xylem and phloem, they become the root, stem, and leaves. So these evolved later. So to give rise the majority of the plants at that particular point in time, the ferns, yeah. So when the dinosaurs was around, ferns popped up. And what ferns don't have are seed. Seed is evolutionary adaptation, allows the plant embryo to only germinate when conditions are favorable for survival. So definitely very important evolution. So ferns are seedless, okay? They use spores. So the spores are tough haploid that allows them to disperse to long distances. So they're very tough. So they're seedless, they use spores, and then do have vascular tissue. So the next one to branch out will be the gymnosperms. So the gymnosperms, they do have seeds, but the seeds are exposed. They're not enclosed in a fleshy fruit. So these ones are your evergreen, if you have them at home. You have the pine trees, the cone-bearing conifers. So they have needle like these. They live in taiga forests like Canada. And the last thing to evolve, which is one of the most important adaptations that plants acquired, is flowers and fruit. Yeah. So flowers have both male and female reproductive structure, which turns into fruit. So that is how we get the angiosperm. Okay. So know a little bit of those, not too much, but know some of these terms. Okay. All right. So with that said, let me get back. Okay, let's look at number 4. While there's only one species of Galapagos island tortoise, so they're huge. These are 100-pound tortoises. And the question is, larger island with more red highland have lush vegetation near the ground. So vegetations that are low and lush near the ground, they have a high domed. The idea is that they can't really raise the neck to reach higher ground for food. They don't need to, right? So they have short necks, which restrict upward head movement. They also have short limbs. They are the heaviest and largest of the subspecies. Now, smaller, drier islands inhabited by tortoises with longer necks and limbs with shells that are elevated above the neck allow them to browse taller vegetation. So this is actually natural selection at its work, right? So the same species of the tortoise in the Galapagos, now they obviously are different physically as well. Based on information, which of the following is a plausible explanation for the ancestry? Well, the answer says D. Individuals with different adaptations in shell shape, that's what they talked about, and also the leg length, best exploit the food resources, and they survive and reproduce and left more surviving offspring. So they evolved essentially, yeah? Okay. Now, if you have, if you chosen any other ones, you can always just call it out. Don't worry about interrupt me, or you can send a chat. So if you look at C is wrong, just garbage. They will throw in a lot of possible correct answers, but that's not the best answer. So for example, C is wrong. Let's say random mutations coupled with inheritance of acquired characteristics. Now, first of all, inheritance of acquired characteristics is never correct, it's false, right? And mutation just doesn't make sense. It's possible, but it's not, right? Okay, so anyway, so you can see, and then B is also garbage, right? Tortoise with shorter leg and necks, they're easily preyed on as young animals by rats. That was introduced. There's no mention of rats, so you cannot make that kind of assumption, yeah? Based on information, do you see that? You have to be based on the information given. All right, so number 4 is D. Let's look at number 5. We'll get to those questions quickly. Number 5 is not too bad. The graph above shows the distribution of beak depth in finch population. So we're talking about finch, right? So you can see the deeper the beak, the probably longer and larger, and they will specialize in different type of food, okay? All right, and then it says, during a subsequent drought, small seeds normally eaten by the finches were less available. Most of the seeds were large that could be eaten easily by finches with longer beaks. So we're told that because of the environmental changes, the longer beak, the one that has 9, 10, 11, maybe 9 and 10, are now the most adaptive one, right? So the question is, which of the following predicts the distribution of beak in the finch population? So there's going to be a directional selection. Natural selection, there's 3 types. Directional selection favors the long one, which is the example here. There's also stabilizing and disruptive. So in this case, then the only directional is A. So you're favoring beaks that are deeper than 8, 9, and 10, right? 9 and 10 was the average here. Very few that were actually 12, 13, 14. But when there's a drought, 12, 13, 14 are selectively selected for, and they're more competitive. Yeah, so that's the only one. Okay, if you look at D, D is more like disruptive. So you're favoring both extreme, right? All right, so anyways, I just want to point that out. So 3 type of natural selection. All right, 6, 7, 8, 9, 10 here. So the 5 questions here is the data in a set question. So make sure you get those, guys, yeah? So this one here, if it talks about, it's clear enough, yeah? So researchers investigate the habitat preference for 2 species of garter snake. So we have this TS and TA. So 2 species, you don't have to try to pronounce them. To create a choice chamber. Now, choice chamber is one of the labs that you do need to know. It is essentially, I think the choice chamber, usually the setup is like this. You have 1 chamber on the left side, and then you have the insertion point right here in the middle, and you have another chamber on the right. So this is just an example of a choice chamber. Okay, so choice chamber, that means you're giving the organism that are inserted here 2 choices based on, for example, the light. So one of them can be light, one of them can be dark. Only 1 variables are altered. One of them can be, in this case, humidity, water, and the other one is no water, right? So again, you can only vary 1 experimental variable. Okay, that's the choice chamber. Yeah, so it is a lab. You need to know. So to create one, we build this enclosure. You can see here. So 1 is in the pond. So you get drier as you go 1, 2, 3, 4. So 4 is the furthest away from the pond. So this is the preference for water. So 2 snakes. They're different species because you can see the same genus. They're both T, right? Different species. So the question is, in series of experiment, researchers induce 1 snake, a single snake. So we should highlight that when you do this, right? Enclosure at 7 a.m. The researcher recorded the location of the snake at 6 time points throughout the day. So we want to see, is it at 1, 2, 3, or 4 to see what the habitat preference is for the snake. Okay, and then in a different experiment related, you introduce now 2 snakes, 1 of each, into the enclosure at the same time, and observe the location of each of the 2 snakes at the time point. So this is what they found, right? So if you have TA only, they prefer 1 and 2. Again, reminder, 1 and 2 is close to water, right? And if you introduce S only, TS only, they also prefer 1 and 2. Okay, even though different times of the day, but 1 and 2. And if you look at the related experiment, now you have 2 snakes, TA and TS. And looks like the TA is now 3/4, and TS is still 1 and 2. So to me, this seems like TS is more competitive for 1 and 2. So it kind of pushes TA out of its fundamental niche, right, into the realized niche. So the realized niche is only a portion of what they can potentially occupy. So it's a fraction, a subset of the fundamental niche. This is competition then for resources. This is not predation, this is competition for resources, okay? So again, TS is more competitive. With that in mind, let's look at number 6. Based on the data, which of the following best describe the habitat preference of TA when introduced alone? So now with the other guy, well, TA prefer B. Preference for zones in, which is 1, or near water, which is zone 2. So they prefer 1 and 2. And the data is supporting that here in the first column, 1 and 2. So number 6 is easy, just reading off the table. Number 7, Number 7, based on the data in the table, which of the following best predicts the result of a study in which the natural population of TN and TS are observed together in an environment that includes freshwater pond? So basically, this experiment, if this were to be found in the wild, what would this look like? The answer is C, right? The TS, which is more competitive for water, will be observed near the water, whether or not TA is present. And that is supported by experiment 2 and experiment 4, the second column and the fourth column, actually. Does that make sense? Yeah. So second column by themselves, 1 and 2. Together, they're also 1 and 2. So regardless, they are more competitive for 1 and 2. All right. So that's number 7. Let's do number 8. Right. Which of the following additions to the experiment design will help to test whether the observed habitat preference will the result of competition between the species? Okay. So if you look at choice A, for example, yeah, placing 2 individuals from the same population together inside the enclosure, and B, introduce different type of plants. Okay, so B is a toss away because we don't know what the food preference is, and the first thought is that they don't eat plants, and plant does not influence them. So you're looking for competition between species, right? Okay, so you don't want to, plant does not compete with snakes. So doubling the number of repetition for the experimental treatment for TA alone. If it's TA alone, I don't see competition as much, right? So it looks like number A is A. So again, you want to see if the test habitat preference was the result of competition between species. So you put individuals. You basically what you did the guy did here. You put them in there and see if they compete for something. In this case, they compete for water. Yeah, and the time repeating that's long is so the effect is you need more than 1 individuals to compete, right? So that's the best answer. Sometimes you don't see a favorable answer. So yes, I might be lagging because it shows up as I'm unstable, okay, or internet is unstable. All right, so let's look at the next one, number 10. Both species of garter snake prey on newt, which is an amphibian that produce a potent neurotoxin. So again, the toxin is a chemical defense that preys have, right? If you're a prey, what kind of defense would you want? Okay, if you're a prey, you want to protect yourself. So there's physical defense, like the thorns or porcupine hair. There's chemical defense, such as example here, neurotoxins. If you eat it, you're gonna probably die, right? Chemical defense. There's also behavioral defense. So the behavior defense, you can think about them as what is the behavioral one? Maybe this is a little bit loosely speaking behavior, but you also have like camouflage, right? You can also do mimicry. So camouflage, blend into the environment. You can have mimicry. You can resemble another organism that are scary, right? So you kind of pop up and scary, right? There's a lot, right? So there's a lot of these behavioral mechanisms. So that's the way, whether you're a plant or you're animal, to defend yourself against. So this is a neurotoxin, which is great. So however, neither species of garter snake is affected by this neurotoxin. So maybe that's why they prey on them, right? Because they don't care. So they're reciprocally evolved to handle that kind of toxin so they can eat it. Yeah. So the resistance to TTX is associated with the mutation of the gene. So that means that both garter snakes are mutated, are mutant, because they're resistant to this toxin, right? Which of the following best supports a claim that TTX resistance arose independently in their in both of these? Now, what that means, guys? Let's say this is the TA phylogeny. Okay, let me use TA snake. I'm going to use a little a, the garter snake, and this is the TS garter snake, right? So if you look at the evolution like this, okay, like that, and maybe they were once the same species like this, yeah. So if they evolve independently, it will look like this, guys. Independent evolve, whatever X is, okay. If they're not independent, they would have evolved in the common ancestor that gave rise to both type of snake, yeah. So TA and TS. So this is not independent. I don't know how to call it, but this is evolved. You know, they evolved so that everybody has it. Okay. So this is not independent. So this is evolved not okay independently. Yeah. Right. So like that. So which of follow? So you want to support the claim that they do evolve independently. Number 10. Okay. A says the stomach contents of the fossil food item contain both DDX. That means they ate it. We already know that. They ate the newt. That's why they're DDT in the stomach content. I don't think that helps at all. Yeah. B, both species possess the gene. Not good enough because you have to have mutations in this gene to be resistant. So not good enough. Okay. And C, this would be the best. That the two species have different mutation. Not the same. If they're the same mutation, they're not independent. But if they have different mutations, so for TA, maybe the mutation occurred, okay, but like a point mutation. Maybe the TS had a mutation that is friendship mutation, but that is independent, not due to the same event. Does that make sense? Yeah. So C will make the most logical answer. All right. Let's look at the next one. Okay. Number 11. Which of the following statements best describes how a growth factor stimulates cell division? Now, growth factor is a type of hormone. It is released usually by injured cells or other parts of the endocrine gland to stimulate cell division. Yeah. So a cell receives this growth factor will start to do mitosis, enter the cell cycle, for example. All right. So what is the how a growth factor stimulates cell division? The best answer here, 11, is B. The growth factor bind to the receptors on the cell surface, trying to talk to the cell, initiating a signal transduction pathway and leading to the activation of a gene or turning on certain specific enzymes. So that is B, signal transduction. This is how signal transduction works, and that's how growth factor works. Yeah. Okay. So most of the hormone works this way. So make sure you know receptor binding, signal transduction involves secondary messengers and cascade of reactions, and then finally lead to cell response, which is the turning on of a gene. Yeah. Okay. That's 11. 11 is B. Okay. Next one. Alrighty. So this is a cladogram phylogeny. So this is similar to what we were doing actually. I'm looking at this and you see that we have vascular tissue that has to do with the plants, right? So some of them have vascular tissue, some don't. Some of them have seeds, some don't. And some of them have flowers. So isn't that the three derived characteristics that we have here, right? If you have vascular tissue, you have seeds, you have the flowers, yeah? Okay, so anyways, the question is number 12. The table above shows the presence or absence of three derived characteristics, cladogram here, for several selected plants. The cladogram below was created based on the information in the table. Okay, so there's a bunch of species, right? Okay, so group 1, 2, 3, 4. Okay, so group 4 of the cladogram, so that would be the last one right here, most likely include which of the following species. So group 4 has everything because flowers have evolved for them, because seeds have evolved for them, because vascular tissue have evolved for them. So 4 possess everything. So you're looking for organism now, going back, who has everything, right? So number 12, the answer is A, right? So A is PS, okay. So P has everything and S has everything, right? That makes sense? Okay, so you just have to read out the cladogram, who has everything, right? So P and also S. Now that means they can tweak the question and ask you who is group 1. So who is group 1, the poor things that don't have vascular tissues. They barely made a plant. Okay, so they don't have anything. So you can see group 1 will be Q, they have nothing, and T, they have nothing, right? Very sad. Okay, they are small, they live near water, then they can't really grow big. Yeah. So the answer is A for this one. So know some of these categories, they're really easy. All right, so here's an ecology of food web, which is the easiest type of question. You cannot miss any kind of food web. Number 13, you don't have to read too much of this, but they give you these data tables. Well, they give you the graph, the chart, so that sometimes you may use it. Okay. So the question is, which of the following changes to the environment will likely lead to more energy entering the community? Well, the more energy enter, the only way energy can enter is via plants, right? So the sunlight, the energy, photosynthesis, right? So which of the following, right, will likely lead to it? Number 13 has to be C, increasing the amount of light, because that helps the plant to do photosynthesis, right? Taking away the squirrel, the total energy is still the same. Nesting site for hawks doesn't make sense. If you apply chemical pesticide for spiders, no, yeah? So those are too far stretched, okay? But C is the most direct answer, okay? All right. You could argue that if you kill the other organism, there's more plant. If there's more plant, they can do more photosynthesis. But that's not the simplest explanation, okay? It's like saying that your friend didn't come to school. Maybe he's not feeling well. Instead of guessing that he went to Hawaii and then he is currently on the beach, too much difficult. So always go for the simplest answer, okay? Don't think too much. Always the simplest answer is usually the best. All right. Another data in the set, okay? So let's take a look at this one. We're talking about a type of fish. It says, let me stretch it out a little bit more. This data in the set, I'm afraid of data in the set questions on the test, yeah? Because there's so much information and it feels like, do I have to memorize everything? The answer is no. So what you want to do with these questions is that as you read them, try to underline or circle whatever is important, because they are there for a reason. They are clues. You have to pick up those clues. You can't do those questions unless you recognize the clues, yeah? Plus the clues, you also have to use your own concept that you learn. All right, so let's take a look at this one. So the fish is found in both marine and freshwater. Great. The marine fish have no scale. So marine have no scale. I need that. But they have hardened armor plates. So they don't have scale, they have plates. The plates are what protects them from predator. That makes common sense. So here, what do we underline? Probably the plates, yeah? So marine fish have no scale and they have plates, yeah? In the late 1980s, Steinbach from the marine population colonized the lake. So they moved into the lake, a freshwater lake. Okay, so it's not marine. Researchers sampled fish from the lake. So this is the data. It's the fish from the lake, right? Every 4 years and record the armor plate. Remember the armor plate was found in the marine, right? So if you look at this, starting from the first time they sampled, 1990s, there's armor plate is low. And over the time, you see there's more and more armor plates, right? So presumably the marine fish migrated to the lake, the freshwater. So with armor plates coming in, there's more and more and more and more armor plates. Yeah, so that's it. So these are low armor plates. These are complete armor plates. Actually, let me see, I didn't read that. So low, partial, complete armor plate. So there's 3 types of armor plate. All right. The armor plate phenotype will categorize either complete, partial, or low. So I guess the complete one is the best protection for predator, right? So it looks like the complete one is dropping and the low ones are increasing over time. All right, so we know what is selected for here. So if I would ask you, what is the best phenotype for lake? Then you have to say is the low armor plate. That's the best phenotype selected for. That's why there's most of it, right? There's no selective advantage having complete armor plate or partial armor plate. There's a low armor plate that is completely favored, almost 90%. Yeah. All right, let's see. So number 14, which is an independent? So sometimes when I look at the question, I still can't help myself to draw X and Y. And then so I don't want to make a mistake on this. So independent would be the x-axis, independent variable, because y depends on x. So y is the dependent. So I'm looking for the x-axis, and that will be the time, right? So 1990 94, 98, and so forth. And phenotypes are the y-axis. Okay, so number 14 is A. Let's number 15. To evaluate the reliability of the results, it would be best to know the following. So now we've got to critique this a little bit. Now, don't try to come up with your own answer, because who knows, right? So you got to read it and eliminate. So this is an eliminating question. The surface area, do I need to know that? The number of male in each group, do I need to know that? Daily temperature, do I need to know that? Age of the cytobag. So what do I need, right? Okay, so how do we know this for sure, right? Okay, so for example, it says here, percent of male in the sample. What if they only sampled 10 fish? And that means 1%, 90% of 10 is 10 fish only. What if they sample 1,000 fish? Then 90% of that is quite a bit, right? 900. What if they sample 10,000 fish? You need to know how many fish, because the percent wouldn't make sense for a small population. That is not reliable. If you only sample 10 fish in the whole lake, as opposed to you sample 10,000 fish. So you need to know how many, yeah? And the male or female don't care, because we are sampling male, okay? But there's a number that makes the percentage more reliable, okay? So this has to do with that. All right, let's look at next one. So 15 is definitely B. Number 16. Now the same question here. So the question is, which of the following graph best represent the type of selection? Again, there's 3 types of natural selection. Stabilizing favor the median trait, like the birth weight for humans, not too big, not too small. And also directional or disruptive, okay? So number 16, what kind of selection is this? Okay, this would be a D, directional, yeah? So why is it D, not B? So you have to look at the key. You see that solid line is before selection, dash is after, and then the arrow is the selection against. Who are we selecting against here? You see that there's 4 low, complete and partial. So we're selecting against complete and partial. So that means the arrow, which is the selection pressure, okay, should be on the large one, right? So you see that? So this is the best one. So you're selecting against the high armor or complete armor. And then so this is before and this is after. Dash line is after. So this is the best graph. Does that make sense? All right. So you got to look at the key. Otherwise, it looks kind of all the same. Okay. Let's look at number 18. Okay. So that's a new question now. Great. So we'll finish data in the set. Now, data in the set does get harder and harder, guys. They will start usually with questions that requires you to kind of read the data and retrieve information, to usually towards the end, application question on evolution and mutation. Those are hard. Okay. All right. Number 18. Intact cells of 2 unknown cell type were placed in solution of different concentration. Okay, so you put into sodium chloride, like salt water. Type 1 swelled and burst in a solution with the lowest concentration, like an animal cell, but there's no cell wall. Type 2 swelled, but did not burst. So my first guess is type 1 is animal cell, type 2 swelled up, maybe it's the plant, right? We don't know. Yeah. So which of the following description of type 1 and type 2 are most consistent with the data? So that's easy. So number 18 is A. 1 is animal cell. That's why it's swelled up and burst in low concentration. That means lots of water. And type 2 must be a plant. So there's a cell wall. So that's why they don't burst. So that's the answer. Okay. Next one. Number 19. In an experiment, scientists isolate mitochondria. Now, first thing popped to my mind, mitochondria is used to be a free-living aerobic bacteria, right? It has its own DNA. Mitochondrial DNA are passed from mother to every children, yeah, son or daughter. All right, but okay, so now you put them into 2 buffer solution. One is at pH 4, one is at pH 9. The scientists found that the mitochondria in pH 4 makes ATP. So pH 4 means more acidic, that has lots of H pluses, and that is probably required for ATP synthase. Remember chemiosmosis? chemiosmosis is how mitochondria electron transport chain makes ATP. So you need a lot of protons for proton gradient. But those in pH 9 did not. Okay, that kind of makes sense. That's my hypothesis. The results of the experiment can be used as evidence to support which of the following scientific claim. So these kind of questions, you got to read every choice, yeah, to figure out, to eliminate. A, mitochondria in free living environment are unable to convert thermal energy. Nobody can. Nobody can convert thermal energy. Thermal energy is lost to increase the entropy of the universe. The second law of thermodynamics, you cannot convert thermal energy anywhere. So that's just garbage, fundamentally false. And B says that electron transport chain pumps electron from cytosol to mitochondria. Well, this never happens. Electrons are never being pumped. It is the proton that are pumped. Electrons are transferred and passed along the membrane proteins on the electron transport chain. So you're not being pumped at all, never, ever, yeah? So that's just a wrong thing. So you eliminate 2, garbage. So you have 2 choices left. Okay, C, ATP production in mitochondria require hydrogen gradient. Yeah, that favors movement of protons into mitochondrial matrix. Well, that sounds good. So I'm gonna keep it, and because this is the elimination question, you gotta read the last one. ATP synthase molecule change their orientation in relation to the proton gradient. Well, that is weird. They're saying that mitochondria ATP synthase looks like this. Now it changed orientation. Maybe it flipped. I don't know. That doesn't make sense. So you want to eliminate those crazy answers, right? So best answer is C. Eliminate 2. I'll always guess if you're not sure. Yeah. You cannot leave any question blank. There's no penalty. Yeah, guys. Okay. So never, never, never leave it blank. That's the worst thing to do on AP Bio test, is to leave anything blank. Number 20. Which of the following best represents 2 signaling pathway that shares a second messenger? 2 signaling pathway is the key, right? So this is one pathway, right? I only see 1. Here I see 2. Yeah. Okay, so we have to see what the best answer is. So this one you got to go through the choices and see which one makes sense, which one does not. Okay. 2 signaling pathway that share, oh yeah, then the C is the best, right? Because you have 2 different chemicals, this one and this one, and they share the secondary messenger. Second messenger. Now the purpose for second messenger, don't forget, is to amplify the original chemical response, right? So the original, the primary messenger is actually the hormone or the growth hormone. You only have a few. But you can produce lots and lots, or maybe 100 times more secondary messenger. Each one of them can trigger the cascade reaction, can amplify the signal transduction, leading to very fast and very quick, very strong cellular response. So that's the purpose of secondary messenger. So make sure we understand that. Okay, so I want to make a note of that. Why do we need secondary messenger? Okay, so these ones are. If you look at our notes, you can just go view the notes. Okay, they should be there. Yeah. So we talk about signal transduction. This is the third time we see this kind of question in application. Okay. So signal transduction is so important. That's how our body communicates internally. Signal transduction. Okay. So what are the 3 steps again? So again, receptor binding. Second one is the transduction. And transduction involves, again, always the secondary messenger, which amplify primary messenger. Yeah, primary messenger. The primary messenger is always the original hormone. Okay, and don't forget that when you look at these kind of questions, only the protein hormones use signal transduction, right? This way, because steroid hormone, they don't do signal transduction. They enter the cell and they bind to DNA directly. Yeah. Okay. And the third one is the cell response. Okay, so let me put that here. This is the cellular response. Cellular response can turn on gene or enzyme. That's it. So this is used by protein hormones. Okay, so protein hormone. Most of the hormones that we have are protein hormones. So it's important to keep that in mind. So if you're talking about the steroid hormones or lipid hormones, they don't do signal transduction. These one will enter nucleus. So, well, first of all, they enter the cell, enter into cytoplasm, cytosol. There's a receptor there. So there's an intracellular receptor. In the cytoplasm. And the whole complex enter nucleus and bind DNA. Enter nucleus and bind to DNA. That's the only way they can work. There is no secondary messenger. So these guys here, there's no secondary messenger. There's no signal transduction, no cascade of reactions are turned on. Okay, and they enter the cell. And where the top one, the protein hormone, they never enter the cell. Okay, they do not enter cell ever. They're too big, too polar. Proteins interact with the protein, the membrane receptor. Do not enter cell. Okay, they bind to membrane receptor, and many use signal transduction. Yeah, many use, well, all of them use signal transduction, but many use a secondary messenger. So they use signal transduction. Okay, signal transduction. Again, it's just chemical reactions that turn on one enzyme after another enzyme, a bunch of enzymes, bunch of reaction gets turned on. So this fundamentally a little bit different. Okay. So let's look at the next one. So this is another time we see signal transduction. Let's look at 21. 21, we talk about genetics. I don't like genetics at all. Yeah. So eye color in a particular strain of fly is influenced by one gene with two alleles. Every gene has two alleles. Yeah. Unless you're multiple allele, you have 4, 5, 6 alleles, but you still pick two. One from mom, one from dad. So here is a simple dominant allele that is red. So a question like this, I always write it down. Red. And then the other one, little r, is a sepia, which is not red. Red-eye female from true breeding, that means homorecessive. Okay, so think about simple example first. I'm gonna imagine this is just regular autosomal recessive. Yeah. So my red female will be big R. Mated with sepia male, so little r. So don't think about sex link. Always with the simplest one first. Yeah. The F1 offspring are all red, and that's good. Okay. And allows you to breed, produce F2. So what is F2? Two F1 by default cross with each other. So based on my original assumption, this will give me 3:1 phenotype ratio. 3 will be red, 1 will be sepia. All right. And this is the case here. Red, sepia, red, sepia. So do you see whether you're male or female, you are 3:1. That means it's not sex linked. It has nothing to do with gender. So whether you're male or female, the ratio is 3:1. So even though this is autosomal recessive, they still introduce male and female to confuse you. But when there's no differences in the female ratio and the male ratio, you can ignore the possibility of sex linkage. It is not sex linked. Okay, so the question is, which of the following best describe the likely mode of inheritance? Autosomal recessive, right? Okay, so it's not sex linked. So autosomal have similar phenotype ratio. So what do you think? The answer 21 is A. That's what we said. The male and female have the same ratio. If the male and female have different ratio, then it's gender based. Then you got to consider sex linkage. Yeah. All right, that's it. So A is the answer. You don't have to look at the other one. Yeah. Okay, another data in the set. So here you have 4 questions about Tay-Sachs disease. So it is an autosomal recessive allele. So we already told this is not sex linked. This is autosomal. So the gene located on autosomes. And this is the hexa gene, affect individual exhibiting severe neurological symptoms that do not survive to reproduction. So they cannot pass on to their offspring. They die, right? Okay, if they're diseased. So right away then, we have, I guess, little t is disease. And they will die, yeah? So big T, little t, this would be normal. And the heterozygous is the carrier. Individual who inherit one copy typically show no symptoms, so confirm our assumption, they're normal. The frequency of Tay-Sachs carrier, so we're talking about the carriers, yeah, right here, the carriers. In the general population, in 3 different subpopulations are represented. So general population is very low, and this is like 37, 35, 20. General population is only 4, okay? Number 22, let's see what they want. Okay, so keep that in mind. Based on the information represented, which of the following best explain the difference in phenotype between Tay-Sachs carrier and homozygous recessive? Well, phenotype, homozygous recessive is dead. They have neural disorder, right? And Tay-Sachs carriers are heterozygous. They're normal, right? So 22, why is that? Well, because having one dominant copy is enough to produce no symptom. But if you don't have a dominant one, then you have symptom, then you're disease, right? So that's the reason, okay? Because having a big T, the dominant allele means that you still have the DNA that is normal. You have the normal DNA. Okay, so 22, let's look at the first one. Tay-Sachs carrier received a vaccination. That's just garbage, right? So there's no mention of vaccination. Number B, Tay-Sachs carrier inherited extra chromosome. Doesn't make sense. This is autosomal. This is not like Down syndrome or Turner syndrome. Yeah, you don't have abnormal chromosomal number. And Tay-Sachs carrier have access to a critical nutrient that homozygous receptors do not. That doesn't make sense. It's not the food. So, and the answer is D. The Tay-Sachs carrier, they are able to synthesize an essential enzyme to have no Tay-Sachs disease, but homozygous recessive individual cannot. Well, this is the assumption that the DNA that they have, the big T allele allows them to produce the essential enzyme so they're disease-free. And if you're little t, you're probably mutated or whatever reason, you don't have that particular gene expression, so you don't have that enzyme, and you will have Tay-Sachs. So D is the best answer. Number 23. Researchers claim, so these claims are difficult, guys. You have to, claim questions are not easy, yeah? Okay, so 23, let's look at this. That Tay-Sachs carriers are protected against infectious disease, TB, tuberculosis, yeah? Those TB skin tests that some of you guys have done some time ago. So, which of the following observation, okay, again, we're saying that the carriers are resistant, okay, to TB. Which of the following observations about annual incidence of TB in subpopulation 2 could be used to support the claim? Well, subpopulation 2 has lots of carrier, relatively. 35 is the highest of all the populations, you see? Well, actually not 30. 35 is pretty high. 37 is the highest, okay. So we're saying that, so we want to, what do we want to do, yeah? Support researchers' claim, yeah? Okay, that, what is this claim? That carriers are resistant to TB. So will A do the job? So you got to read it. This you got to read everything. That's why it's hard, yeah? And you have to eliminate it. There's another way. So the incident TB in subpopulation 2 is roughly equal to incident TB in the general. Well, that is against the fact. That's wrong. The general one, if you see, is only 4. 2. Subpopulation 2 is 35. They're not the same. So this is just something that is tossed away. So this is a giveaway. Okay, next one. The incident TB in population 2 is greater than incident TB in population 3. Well, that is a fact. So I'm going to keep this one and come back to it later. The incident TB in subpopulation 2 is lower than incident population, okay, in population 3, right? Okay, so here, we talk about the carrier here, right? So this is the connection here that even though the data is showing us the carrier, but we're talking about TD. So it's between B and C. One of them is, they're like kind of the opposite. Okay, remember the claim is that carriers are resistant. So if you're resistant, wouldn't this have less TB? Wouldn't this have more TB? Because the population 2 has more carrier, so there should be less TB. This would support the claim that carriers are resistant to TB, right? So do you see B and C? B is wrong because the incidence of TB is greater in population 2. Wouldn't that suggest that the opposite? Okay, so it has to be C. The incidence of TB in subpopulation 2 is lower than the general. So the more carrier, the lower the TB. That is what the claim is, that carriers are protected against TB. So between B and C, they're kind of opposite. So usually one of those is an answer. Yeah. Okay. All right. So that's the answer. C is the answer. Yeah. All right. 24. Which of the following ethical questions about Tay-Sachs disease cannot be answered using scientific methods? Okay, so this one again, you got to read through everything. It's a lot of reading. With a difference in the allele, remember this is the allele that they talk about, the gene, right here, the gene that they mentioned earlier, right? Let's review what the gene is. So it says a disorder caused by autosomal recessive allele of this. So this gene. Okay, so if you're recessive for this gene, you have the disease. Yeah. Okay, so that's the gene. Okay, let's see. So what is the ethical question that cannot be answered? Okay, so let's look at 24A. Would a difference in the hexa allele in subpopulation 1 and 2 affect the severity of the symptom? So you can actually look at the patient in subpopulation 1 and 2 and see who has more severe. I think you can answer that. B, should genetic testing be required before individual 1 before individual 3 are advised to not have children? So you're telling in population 3, you tell them don't have kids. That is not ethical. You cannot tell people don't have kids because you have genetic issues, right? That would be a form of discrimination. So 24 is B, yeah? Okay, well, let's read on. Again, you cannot just based on people's genetic and discriminate them because they have a tendency to have children, right? If you have 2 carriers, you could have children who are diseased, not to have children. You can't do that, okay? So B is the answer. C, could the frequency of the carrier be a consequence of genetic bottleneck? But genetic bottleneck means genetic lack of genetic variation. Bottleneck effect produce lack of genetic variation. So you could find out, is it because the population doesn't have enough variations? Maybe they're all carriers that a lot of carrier for some reason for this part. So that is not unethical. Okay, so that's fine. Okay, anyway, so B is the answer. Okay, that would be the best answer there. Okay, next one. So you cannot. Okay, 25. We have a pedigree. Yeah, if you really don't like pedigree, don't skip it. Yeah. Okay. So the question is, which of the pedigree represents a family with a history of Tay-Sachs? So again, affected are shaded. Okay, and circle is female always. Yeah. So if you look at this one. Now, why is this wrong? Why is this wrong? If you're affected, we're told they die, right? They do not survive to reproduction, so you can't have this, right? So you just can't have that. Where is that part about they don't survive to reproduction? It clearly says that, okay? So that's why if you read these, sometimes you need to look for it. So right here, right? They do not survive to reproductive age. So you can't have diseased male and female and then have babies with each other. So by elimination, I already crossed everything out. So the only one left is B. So that means that the only way to have this family history is to have carriers. They're normal, they don't have the disease, but boing, they have killed children that do have it. Very sad. Yeah. And that is how majority of disease are passed on. Vertical transmission, right, from mother to children. Okay. Using a hidden recessive disorders. That's how permissive they are, persistent they are. Okay, that's how they passed on. 26. Which of the following processes is most likely to occur as a result of animal cells. So this is a different question, yeah? Animal cell have a signal to apoptosis. So you have to know what apoptosis is, right? Apoptosis is a good process that causes a cell to die on its own. It's like cell program, it's a programmed cell death, right? Usually the cell is old, worn, DNA is damaged, the potential for mutation is too high. You don't want them to stay around, so they will signal its own destruction by apoptosis. So good process. So this is easy. Just know the term. 26 is C. You trigger the creation of these enzymes released by lysosome self-destruction. Yeah, has nothing to do with making protein. Has nothing to do with growth factor, right? So not a vacuole. So lysosome intracellular digestion. Yeah, digest itself and die. 27. In mammals, mammals have hair. Mammals have mammary gland that can produce milk. Mammals are like marsupials, like platypus, like placental mammal, like us. Okay. Now, mammals, an increase in concentration of sodium. So what happens when you have a lot of sodium in your blood? Common sense, guys. If you have a lot of sodium, you eat too much salt. Water will go into your blood. That's simple osmosis. Your blood pressure will go up. And this is why people with heart problems cannot eat salt. If you feed people with heart problems salt, like grandparents, like Takis, those very salty junk food, you're going to kill them because the blood pressure will shoot up and that can cause heart attack or aneurysm or whatever. Yeah. So it's dangerous. So that's what this means. So you're thirsty. Thirsty also, you lose so much water, your concentration of sodium in the blood is high. So either way, this means that salty conditions. Yeah. So you'll release this hormone, anti-diuretic hormone, to counter that. Okay, so let's say we have no knowledge of anti-diuretic hormone. What this hormone does is for you to pee less, to conserve water. Why? Because you have too much salt. You need water. Okay, this means that usually this means lack of water. When you have too much salt, okay, your body will try to increase water, retain water. So you pee less, you release anti-diuretic hormone. So water will now be able to go in to your blood vessels. But the consequence of that is shooting up blood pressure. Okay. So as the concentration, so you don't need to know this because they will tell you. As the concentration of sodium returns to this regular level, ADH is reduced. Isn't this negative feedback? Right? So high sodium, you release ADH. And when sodium level drops because they're being diluted by water that you retained, ADH level drops as well. So negative feedback, yeah? So what is the role of ADH in maintaining blood concentration? Osmolarity is a fancy way to say concentration, yeah? So you see this ADH promote increase in the movement of sodium. Now that is counter to what was said, right? You have high sodium, you release ADH, so your sodium level drops. That is this is opposite of what the information says. B, ADH promotes the increase in movement of water into the bloodstream. That's probably the best answer because that's how they dilute blood osmolarity, by getting water into the blood. Yeah. And it is not C, you don't want to excrete water. If you excrete water, your blood becomes more salty, sodium concentration will be even higher, right? Okay, so the B is the best logical answer. So you should know anti-diuretic hormone, yeah? Anti-diuretic hormone causes you to pee less, so your kidney reabsorb more water, so that and it's usually triggered by thirst, exercise, or too much salt in your body. All right, number 28. You have this whole diagram to utilize. So if you look at this, it's actually cell cycle. If you remember, cell cycle begin with G1, it goes through S, DNA synthesis, and then you go through G2, and then you go to mitosis, and followed by very, very quick cytokinesis, right? So don't forget that plant cytokinesis forms a cell plate, animal cytokinesis forms a cleavage furrow, right? That splits the cytoplasm. So this is about cell cycle. And we got a whole bunch of these. If you remember this, that you need this mitotic right here, the cyclin CDK complex. This is called mitotic promoting factor. It's when cyclin combines with CDK. when cyclin combines with CDK, this will trigger mitosis. Okay, so cyclin is broken down right away, then mitosis is all over. Yeah, so you have this cyclin CDK complex. All right. So anyways, this is we don't need to memorize, but that's one example how to interpret that. So number 28. Based on the model of eukaryotic cell regulation shown, which of the following best describes the effect of the drug that blocks production of mitotic cyclin? So where is mitotic cyclin? I'm looking for right here. So now your drug is going to get rid of this. If you get rid of this, then there is no activated cyclin CDK. And if there's no activated cyclin CDK, you're not going to be able to enter mitosis. So these drugs prevent mitosis. Okay? So if you block production of the cyclin, then there's no mitosis. This is your conclusion. So choice A, cell cycle will proceed uncontrollably and become cancerous. Out of control, no. G1, it's talking about G1 function normally. G1 cyclin will functionally replace, but there's another cyclin right here. They're saying that this one will replace the other one. Well, we don't know. There's no information that says that is the case. So, but let's read on. DNA synthesis will be prevented. That is a wrong cycle, right? We are looking at this area here. So the answer will be D. The cell will be prevented from entering mitosis, right? You won't be able to divide by mitosis because that's what this mitotic cyclin is for, okay? So that's the most direct, that's what the picture is. So you don't have to know exactly there's a two type of cyclin, right? But you can answer the question correctly. All right, next one here is about lac-operon. We did lac-operon so many times. And make sure you're comfortable with lac-operon, okay? So I'm just gonna write down some of the key points of lactose operon. And you have to know it, okay, guys? So again, lactose operon is an operon. Operons are found only in bacteria. Eukaryotics do not use operon. The purpose of operon is that it's simple and fast. One promoter can control 3 related genes, which allows, when they are expressed collectively together, allows the bacteria to utilize, in this case, lactose. Okay, so this is one promoter. Promoter controls 3 genes for bacteria to use. That's the whole purpose of lactose operon. Now, because lactose is not the preferred nutrient, lactose operon is not on all the time. It is only turned on under 2 conditions: when there is no more glucose, the preferred nutrient, and when lactose is available. Those are the 2 conditions, right? It kind of makes sense, right? Why turn it on when there's glucose around, right? Glucose does not require digestion. And why turn it on when there's no lactose? So this one is normally off, so it is inducible. It is an inducible operon, so it can be turned on with 2 conditions met, yeah? So number 1, 2 conditions met. The first condition is that there has to be no glucose. Bacteria will just use glucose instead of lactose, yeah? We'll just use glucose. Secondly, so this will conserve energy, okay? So waste of energy to not to use glucose. And second one, lactose must be present. So why turn it on when there's no lactose around to digest it in the first place? So both of these is to save energy, to make bacteria more adaptive, okay? So that's what it is. And if you look at the key players for these kind of questions, you'll see words like, okay? You'll see words like what? You'll see words like, promoter, okay? Operator, you'll see a regulator. Regulator makes a repressor. Regulator gene, they codes repressor protein, yeah. And you'll see the structural genes. So those are some of the terms you'll see, yeah. Okay. So again, promoter is where RNA polymerase binds So make sure we know them, yeah? Promoter is always where RNA polymerase binds to begin transcription. All genes starts with a promoter. All expression, all transcription starts with a promoter. RNA polymerase binds to start transcription. Operator is the DNA where repressor binds. When repressor binds to operator, no transcription because RNA polymerase is blocked. Okay. All right. So with those two, and a regulator gene, you'll see probably, let's see. Okay, let's come back here. So you can see our repressor protein is a circle. Lactose is 2 hexagon. It's a disaccharide. Cap protein, even if you don't know what cap protein is, well, cap protein is there when there's no, well, it tells you, yeah? So let me just, let's read this one. And you have RNA polymerase and mRNA. Okay, let's look at the cap, okay? I talked about cap one time. So let's look at this. So you're told that lactoferrin codes enzyme for the breakdown of lactose. We already know that, right? When lactose is absent, is inactive. Sure, because repressor is attached to the lactoferrin. And then it says when lactose is present, lactose bind to the repressor protein, causing repressor protein to dissociate from the binding site, giving access to RNA polymerase. Okay. And now we'll talk about cap. So it's about glucose. When there's no glucose, which means that the protein cap bind to regulatory site. That's it. That's all you need to know. So you're looking for a diagram when there's no glucose, cap will be there. Okay, and it helps to activate. So cap is good. If there's cap, if there's no glucose, you will have cap, and when there is cap, you'll activate transcription. That's what that means. So do you have to memorize it? No. But if you did, it's easier. But the clue is here, okay? So with those clues in mind, the question is, we want to be looking for a diagram when lactose is available and glucose is absent. So the conditions when the lactoferrin is on, okay? We want it to be on. When glucose is absent and lactose is present. So which one shows that the condition is on, the gene is on, so it's being transcribed, okay? So I need to have RNA polymerase there, so it has to be these two, right? This doesn't even make sense. I look for the easiest one. If you don't have RNA polymerase, how do you turn it on? You can't. So it has to be one of those. And then you're looking for lactose. Lactose bind to the repressor protein. These will attach. Lactose does not bind to RNA polymerase. That's garbage, right? Lactose does not bind to RNA polymerase. Lactose bind to the repressor. You're also told about that. If I may just scroll up a little bit, you can see this portion here. It says lactose bind to the repressor right here. Very clear, lactose bind to the repressor. So lactose bind to the circle, not RNA polymerase, even if you don't know that, right? So the only one, therefore, then, is C. C is the answer, yeah? Okay, so all that review for nothing. You could have just looked at the diagram and then match what you're told, okay? So that, a lot of the questions are like that, okay, guys? Okay, let's look at the next one. Another data in a set, and that means that you want to save a whole bunch of time so you can work on these kind of questions, yeah? So easy ones, like what we did earlier, don't waste time overanalyze. If you're pretty sure, pick the answer, move on, okay? You want to save time for this. So here is about excess intracellular iron. Now, guys, iron is essential, but too much of it can cause cancer, okay? So you can't just pop in iron pill. There are iron pills that a doctor can prescribe, right? Nothing too much is good. So you can't overdose the vitamin. You cannot just, especially those lipid soluble ones, if you eat too much of it, they stay in your system. If it's water soluble, yeah, you can, if you eat too much, you just pee it out. But iron, you can't have too much. It's toxic to the cell. So it tells you the solution. Ferritin is an intracellular iron storage protein that bind to excess iron. You're told what that does. So when you have too much iron, it's toxic, bad. Ferritin, good, when it binds and kind of stores the extra iron. The presence of ferritin can protect the cell from iron-induced toxicity. Sure, so ferritin is good when there's too much iron. That's what we're told. In an experiment to investigate the effect of iron intake on ferritin synthesis, so your assumption is what? If you eat too much iron, then you start to synthesize more and more ferritin. They're directly related, right? So rats were given food containing different amounts of iron. So you can see the x-axis, you have more and more iron, and the y-axis is the ferritin. It starts to have more and more as well, okay? Right? So what does 1 mean, guys? Look at this diagram, guys. 1, it says 1 is because it's relative to control. So 1 is the control. So that means if you eat this much iron right here, maybe 30, there's no difference between ferritin of the experimental group and the ferritin in the control, yeah? But if you eat more than that, like 50, 75, 100, you're going to have more ferritin. That makes sense, right? If you're less than that, you're less ferritin. So your control is right here. That's what this 1 means, relative to the control. All right? So this does support our initial hypothesis that more iron, more ferritin. But what is more? More is more than control. So this would be considered lots of iron, lots of ferritin. Yeah? Okay? All right, so that's what we're told. So the data is not too difficult to interpret. Do you have to interpret that detail? Maybe not. You just want to keep reading first to see what the point is. And sometimes the questions will be separated into 2 parts. So then they start moving on to something else. So it's all confusing. Okay, so let's look at this one. Based on the other data, researchers have developed the following model demonstrating how ferritin synthesis is regulated by iron. So how do you control ferritin synthesis? So protein synthesis, yeah? Transcription, for example. When iron levels are low, there's a repressor of translation. So this is kind of the, okay, so again, you should highlight this. Low iron repressor called IRP, bind to iron response element. What the heck? Which is the stem loop structure near the 5' end of ferritin mRNA. Now, ferritin mRNA makes ferritin. So if you have this repressor, which is IRP attached to the 5' end, then there's no ferritin produced. Look, this is your repressor protein, okay? So I'm going to use a different color. This is your repressor right here. When this repressor wraps around the mRNA, there's no ferritin. And that happens when iron level is low. That makes sense. Remember, we want to have ferritin. We want to have ferritin when there's high iron, right? To store, to protect us from toxicity. But when iron level is low, you don't want ferritin. And this is showing you how. So one more time, it says, when iron levels are low, you don't want ferritin because there's a repressor now. So how does it work? A repressor called IRP, which is this highlighted in blue. What do they bind? They bind to this IRE, whatever that is, but ultimately it is mRNA, the ferritin mRNA. This is part of the ferritin mRNA. This is your mRNA. So when this repressor IRP is attached, no ferritin is produced. But look what happens when iron is present. When iron is present, do you see that? It causes the repressor to fall off, giving access to ribosome in this case, because you want to do translation, you need ribosome. So now ribosome have access, so now you have a ferritin, right? So this is why high iron, high ferritin. That makes sense. This goes with what we are saying. So this works the same way like lactose. Remember, lactose bind to the repressor protein, causes repressor protein to fall off, giving RNA polymerase access to the lac operon. Here, iron causes the repressor to fall off, allowing access of ribosome to the mRNA to make ferritin protein to protect the cell from high iron in the first place. Yeah. So that's essentially what we're told. So a lot of fancy words, but sometimes you read it several times and you read before and after, you can see what is at play. Yeah. Okay, let's look at the question now. Number 30. Based on the data in figure 1. Okay, so figure 1, we got to go back to figure 1. Concentration of iron in a control diet that used the investigation is most likely what? So we're looking for figure 1, concentration of iron. Okay, control. So this is your control. Relative control is 1. So they're looking for this number right here. So this looks like a between 25 and 50. So I'm going to add them. That's 75 and divide it by 2, right? So that's like 37.5. That's my guess. About 37.5. That's what they want, right? So again, usually the first few is just reading from the table, the data, to understand what the table means. So the iron concentration in the control is about 37. So the best answer is, so see, they're very nice. The other ones are way off, right? So they give you a range. So B is the answer, 30. 31. So B is data retrieving, yeah? So read the data, get the answer. And 31, which of the following conclusions about dietary iron and ferritin synthesis is supported by figure 1? So this is also interpreting the data, but it takes one more step. You got to interpret what that means. So to me, high iron, high ferritin, right? So that's what they're saying. So when iron concentration is higher than 37.5, then the rat is producing more ferritin compared to the control. Okay? Control is given, so that means the control rat is given 37.5 iron. And these experimental groups, some of them are taking more iron than the control, some are taking less iron than the control. That's what that means, okay? So you're feeding those rats irons, and the rats that are considered the control group are fed with 37.5 iron, okay, in the diet. So more iron, more, ferritin, okay? So 31, you do have to read all this. So there's no way you can just skip, yeah? So A says, within 31, okay? A says that a dietary iron concentration of 25 has no effect on ferritin gene expression. This is wrong. If you're at 25, there is an effect compared to the control. When it's 25, you have very low ferritin compared to the control. So there is an effect, right? So it's going to inhibit ferritin synthesis. So there is an effect. So A is wrong. Let's look at B. At a dietary concentration of 50, the rat makes twice as much ferritin as the control. Well, let's see. 50. So you got to go to 50 here. Okay. And if you look at 50 here, I am here. Is that twice compared to the control? No, it's 1.25 times that of the control. Twice would be up here, right? So that is also false. It's not what the data is saying. Okay, so far it's pretty easy. So you have 2 left, you guess, right? Okay. 1, occur iron concentration 75 or greater. That looks right. C is the answer. Look, 75 or greater, right here. Seems like you have the maximum ferritin synthesis. So this seems to be the best level. About 75 seems to be the best level to maximize the ferritin production. And it seems like if you keep adding iron, it's not helping. It's kind of plateauing or even going down. We don't know. Okay, that's the answer. Okay. All right. So we have 2 questions. They both require data analysis. Yeah. And D is wrong. It's talking about ferritin absorption from the gut. No. This is. There's no detail on the gut. We're just measuring how much ferritin in the blood of those rats. That has nothing to do with absorption. Yeah. So that's just something made up somewhere. 32. Based on the model of ferritin synthesis in figure 2, which of the following best describe the mechanism whereby iron most likely regulates ferritin production? How does iron regulate ferritin? So here, iron removed the inhibition, right? Iron removed the repressor protein, allows the translation. That's what figure 2 is saying. The presence of iron removes the IRP protein, and allows the mRNA to be translated by ribosome. So iron positively regulates, it stimulates ferritin production. So A, translation occurs under low iron. That is not what the data is saying, right? Because at low iron, there's that repressor. Translation occurs under low ion, so you can eliminate A and B just based on the first statement. You don't need to read further. Translation occurs under low ion. Translation occurs under low ion. No, translation does not occur under low ion, right? So it has to be C and D, high and high. So between those two, you want to differentiate them. If you couldn't, take a guess, right? So let's look at C. Translation occurs under high ion. Why? When IRP ion complex dissociate from ferritin mRNA, permitting ribosome access to ferritin coding. So that's the answer. So again, IRP is this protein that I highlighted earlier in this, IRP. So again, IRP is that protein right here. So when the low ion right here, IRP is attached. And what does ion do again? It causes them to dissociate so that you have translation. And translation is always done by ribosome. So C is the answer. And let's look at what the last choice. What is it wrong? Last one, okay? Translation occurs under high, yeah. And it says when IRP complex bring the 5N closer to the coding region. That's not what the IRP is doing here. IRP simply attach and detach. It does not bring anything closer to the coding. If you really look at that, the distance is the same, right? The coding region is right here. The IRE is here. There's no change in that distance, so that's just wrong. Okay, let's look at the next one. Number 33, okay? So the last two is usually the hardest. So after you interpret the data, now is application. Based on the model of ferritin synthesis, which of the following describe the role of feedback control of intracellular level? So feedback, negative feedback especially, is how we are regulated, how all these are regulated, okay? So feedback control. So you got to read all of them, unfortunately, here, okay? So 33, if you read the first one, decreasing iron activates IRP. Decreasing iron activates the synthesis, right? So usually I read the first part, maybe the first sentence, and eliminate. When I read B, something tells me that is wrong. Decreasing iron activates the synthesis of ferritin. Didn't we say the opposite? Right? Increasing iron, right? Active. So that's wrong, right? Increasing iron activates IRP. Increasing iron. Now remember, IRP is repressor protein, yeah? And IRP binds iron. Okay, so this is garbage too, yeah? IRP does not bind iron. IRP bind to the mRNA. Okay, it's the ferritin protein that binds iron. So that looks off too, right? 33. Okay. So let's look at the last one. Increasing iron activate the synthesis of ferritin. I like that, right? More iron, more ferritin. Ferritin protein in turns. So the final product. When you do negative feedback, it always has to do the final product. The ferritin protein bind to iron. Ferritin protein bind to iron because you want to get rid of it, right? Decrease both free iron and also the level of ferritin synthesis. So again, the final product inhibits its own synthesis. That is what negative feedback is all about. So D is the best answer. Yeah. So B and C is wrong. Let's see what's wrong with A. I didn't read further. Decreasing iron activates IRP. That is also something is off. IRP is usually activated. Okay. IRP in turns activate iron transport protein in the membrane. So there's no questions about this transport protein. So that part is wrong. So the best answer is D. Okay. So we got to analyze them like this. Okay. Let's go to the next one. They force you to read every single choice and eliminate. That is how you answer most of these difficult questions. Yeah. Let's look at 34. After search of nucleotide sequence database. All right, so this is about DNA. Researcher identified an IRE in the 5' untranslated region. So let's look at IRE first. I didn't really focus on that one. Now it's talking about IRE. So IRE is the mRNA right here. So the IRE is this part of the mRNA. This loops up like a hairpin and continue here. So this element right here, IRE, seems to be the regulation point. If the IRE has this protein attached, there is no translation. If the IRE is free, then there is translation. So now this question is looking at the DNA nucleotide sequences of IRE. Yeah. Okay. And then again, this is not IRE is not translated. Only the coding region is translated into protein. Yeah. The IRE is a control region of the mRNA. Again, we're looking at mRNA, not DNA. There's no promoter here. Promoter is found on DNA. This is a piece of mRNA. So the control region is called IRE. Okay. All right. So that was the review for the IRE. Okay. So what about it? It says, looking at the nucleotide sequence database, researchers identified an IRE in the region, right? In the 5' so the left side, the upstream, untranslated, like what we said. Okay, so we found this in this enzyme, aconitase. So we have IRE in aconitase, suggesting that this enzyme, just like ferritin, is also controlled by the same mechanism. So again, IRE is found in the ferritin mRNA, which produce ferritin. Now we found another IRE in another gene coding for a different enzyme, not ferritin. So our assumption is right away that aconitase is also controlled using the same mechanism. And this enzyme is in the Krebs cycle. Great. Question is, which of the following pieces of experimental evidence best supports the claim that synthesis of aconitase is also controlled by mechanisms similar to ferritin regulation? So we want to support this hypothesis. Okay, so then you basically just replace ferritin with aconitase. That's it, right? So IRP bind to the aconitase mRNA in the presence of iron. So this is wrong. Remember, they bind. IRP does not bind when there's iron. Iron causes IRP protein to fall off. So that goes against what the data has been saying, right? Again, IRP is the repressor protein. When there is iron, it falls off. It does not bind. So A is just wrong. Okay, so that will not support it. And B is the answer. The relative amount of aconitase protein increases in the presence of high iron. Well, yeah, because high iron, high ferritin, which means high aconitase if they're controlled by the same mechanism. So that's the answer. And has nothing to do with oxygen. Forget about oxygen, right? And the level of reduced electron carrier has nothing to do with NADH, FADH2. Okay, in the increase in the presence of high iron level. It doesn't make sense. It has to do with aconitase enzyme, nothing to do with NADH, FADH2, even though they are involved in mitochondria. So B is the most direct answer. If you have to choose and guess, it has to be A and B. And A is obviously contradicting what the data has been telling us. Yeah. So B is the answer. All right. So again, these are harder. It really takes a detailed understanding of what's going on. Okay. Let's look at the next one, a different one. So this is about retroviruses. So we should be seeing them this year as well because of the COVID, right? So retroviruses have RNA genome. Sure. They use RNA to make a copy of the DNA. And this is why they call retro. And they need this enzyme reverse transcriptase, right? So this reverse transcriptase will make a DNA copy of their RNA genome. All right, so they are established a latent infection. So they are lysogenic, not lytic, right? By which of the following mechanism does the infection by retrovirus, such as this, most likely cause a long-lasting genetic change to the host? So if they're lysogenic, they actually insert themselves into the host DNA and stay dormant. Every time the host divides by mitosis, they are also divided alone, right? So including the viral DNA and the host DNA, everything is replicated, right? So that is how lysogenic latent cycle works. Lysogenic cycle, okay? So let's look at the choices. A, host cell ribosome translate viral RNA enters the cell. No. Yeah, so if that were the case, that's probably just a lytic cycle. If you start translate the viral RNA, you're making viral protein, you're about to go to lysis, assembly and lysis. No. The viral RNA polymerase transcribes host gene. They're saying this one transcribes the host gene. That's not what the RNA polymerase is doing. Okay. All right. So that's probably a far-fetched. Okay. Let's go to the next one. RNA viral genome is reverse transcribed into DNA. That's what the reverse retrovirus is for. And then integrate into the host. Yeah, that's basically it's describing how lysogenic cycle works. So that is C, right? It's reverse transcription into DNA, and then that viral DNA integrate into the host. That's how you change the genetic changes. Why would that matter for the host? It is a big deal for the host because if the gene is inserted into a coding region, then your DNA is messed up. That gene is altered permanently, right? Of course, that has a consequence on the host. All right, 36. Let's take a look at 36, please. So which of the following best describe the role of mitosis? So the mitosis, the purpose is to split. Don't confuse mitosis with synthesis of DNA. DNA synthesis occurs first, then you do mitosis. Mitosis is to line up the chromosome and separate them so they're identical numbers on each side, okay? So 36, the answer is A. Distributing of replicated chromosome, which is called sister chromatids, right? To daughter nuclei. That's the whole point, right? It does not divide the cytoplasm, that's cytokinesis. Producing organelle, that would be, that's not, the organelles are usually made in the G2 or G1, whatever, yeah? G phase, okay? Exchanging genetic material between homologous chromosome, that is crossing over, that is not mitosis, yeah? All right, so A is the answer. 37, that's easy, right? So 37, the researcher hypothesized that RNA molecules were present in the most recent common ancestors of all living things. Which of the following scientific question would be best to test this hypothesis? If RNA is found in the recent common ancestors of all living things, and 37 has to do with whether RNA evolved first. 37 is B. Do any organism function entirely without RNA? Okay. Okay, it is possible to produce RNA polymer? No. How many? So you have to talk about the evolution, yeah? So A is not to do with the evolution. How many distinct functions can particular RNA? How many different monomers of RNA? Do any organism function entirely without? Yeah, this is the only relevant one. These kind of questions you kind of have to just kind of gauge a little bit. If RNA is found in most recent common of all living things, what if you find an organism that functions entirely without RNA? Then you can refute this, right? Okay, that it is not found in the recent common ancestor. There are actually organisms that are alive that don't have that RNA, right? So that's the most relevant, whether you want to go against the hypothesis or for the hypothesis. All right, let's look at the next one. Next one, we got the cycle moon phase, all righty, and the nighttime high tides through the lunar period. Okay, and this is the day, 30 days. Okay, California, right? Hunting Guaranians is a marine water. Guaranians swim onto the beach and mate and lay eggs. That's when we catch them. So sad. So the researcher proposed that the spawning behavior take place when nighttime tides are the highest. Okay, that means right here, highest right here. Okay. Which of the following pieces of evidence would support the researcher's claim? Guaranians, this you also have to read everything. Guaranian spawning occurs every 2 weeks. Guaranian spawning occurs when nighttime illumination is the lowest. High tide occurs 1 time per lunar cycle, and light intensity of the moon varies with lunar cycle. Okay, so this one is, well, A seems to be the answer because the tide is the highest right here, and that looks like is 15 days or 2 weeks. Okay, and it seems to oscillate the same way, yeah? So that is the only evidence that talk about high tide. Everything else it's kind of weird. It's not the light intensity, it's not about high tide. Okay, so that's that one, all right? All right, that's 38. Let's look at the next one, 39. Okay. Next one here is immune system. All right. So if you look at the immune system, the bacteria, and this is the antigen presenting cell we talked about, right? A bacteria will be eaten by cell X. This could be a macrophage, for example. eats a bacteria and spit it, the antigen fragment onto the surface. That is called antigen presentation. Yeah. And cell Y will be a helper cell, T helper. It gets in the antigen presentation cell present to T helper that this bacteria is invading us. So T helper will activate. This is the B cell. So the B cells have antibodies. So a monoclonal selection will happen so that the B cell will differentiate into plasma cell, which release antibodies into the blood, and this antibody will target. What is the target? Target bacterium, yeah, of this kind. So this is how immune system works. This is the humoral response, yeah. Humoral response involves the B cells, not the cell-mediated response. Cell-mediated response is the T cell that goes after the infected cell. This is the antibody humoral B cell response, okay? So that's what this is. Again, so that's what's going on. Okay, so the question is, the vertebrate immune system, okay, they're wanting to identify what cell X or Y, Z is. So we already did. We said X is antigen presentation, like, and Y is a helper, Z is a B cell. So Y is a helper, that is the only choice is C. And Z is the B cell. So the only choice is C, yeah? Okay, that's it. So understanding that B cells coordinate everything. That's important, yeah? B cells receive input from the antigen presenting cell. They will activate the B cell. They will also activate the T cells. So they activate the specific immunity, the acquired immunity, yeah? Okay, so T helper cell is important. All right. Next one here is talking about feedback. So what this diagram is saying is that your body have two hormones, calcitonin and parathyroid hormone, yeah? These two hormones do the opposite. When there's, okay, it does the opposite, okay? So for example, how do you read this? Because it's a cycle, it doesn't matter where you begin. So let's begin with this, okay? Let me use a pen. So when there's too low calcium, when blood calcium falls, right? Whoops, actually I don't want to begin with there. All right, here. So I want to begin here, yeah? So when blood calcium level is too high, too much calcium, you release calcitonin, which will cause the blood calcium to fall. So that's what this means. Calcitonin lowers blood calcium, yeah? How? How is in the box. You deposit calcium into the bones, okay? And you pee out more calcium, excretion, right? Excrete, waste. So that's why the blood calcium falls. All right. And what happens when calcium level is too low? If the calcium level is too low, you release parathyroid hormone. So parathyroid hormone raises blood calcium. How do I know? Because it says this will lead to a level of calcium to rise. How? By release calcium from your bones. Your bone is a calcium storage. Okay. All right. So that's what they want you to get out of. Calcitonin lowers calcium in your blood to return the calcium level to homeostasis. And when do you release calcitonin? When there's too much calcium. And parathyroid hormone raises blood calcium. When do you release that? When calcium levels are too low. So you didn't have to know or memorize this. That's how this hormone works. Okay. But if you hormones are usually working oppositions, like insulin lower blood glucose, and glycogen raises blood glucose. That makes sense? Yeah? Okay. Alrighty. So the model shown here is the role of two hormones, calcitonin and parathyroid hormone, in maintaining blood calcium level, homeostasis. Yeah. If a dietary change results in increased blood calcium, so if you have too much calcium, you want to release calcitonin. Yeah. You want to release calcitonin because you eat too much calcium, right? So you want to release calcitonin. So A is wrong. Calcitonin level should not decline. So calcitonin will be raised and promote the deposition into bone. So B is the answer. So when you have too much calcium, the diagram essentially tells you that you will release calcitonin. So, and calcitonin lowers blood calcium because in the box it says calcium is deposited into the bones. Okay, so you release calcitonin, which causes calcium deposit in the bones, and that will lower the blood calcium, the desired response. Okay, so B is the answer there. Let's look at the next one. Type 1 diabetes. So type 1 is autoimmune. So your immune cell, your T cells are destroying your pancreas cells that produce insulin. So type 1, there's no cure. Type 2 is usually related to age or stress, right? 41, okay? All right, so a hormone that regulates blood glucose. Again, we usually memorize insulin lower blood glucose. Something with calcitonin, you should memorize. It lowers blood calcium. Okay, so which of the following best explains how treatment with a drug? So you get a drug and it's treatment, so it will help you. So what does it do? It stimulates the production of insulin receptor on target cell, will affect the insulin signal transduction of type 1 diabetes. Okay, so here's what the problem is. They tell you that type 1 produce insufficient of insulin. They don't have enough insulin. That's the problem. Okay, so if the drug stimulates production of insulin receptor on the target cell, that's not going to help at all. But the problem is they don't have insulin, not because the receptors are faulty. Okay, so the drug will have no effect, so either A or B. Okay, again, type 1 is because they don't produce insulin, because they're unable to, because autoimmune. And you're trying to stimulate the production of the receptor, that's not going to help them if they don't have insulin in the first place, right? All right, so it's A or B. So you got to figure out which one is the best answer. The drug have little or no effect, right? And A says, because receptor will not be activated in the absence of insulin. B says, because insulin receptor will not be allowed to enter the cell. What the heck? So which one looks like the answer? Yeah. So 41 should be A, right? The receptor will not be activated. There is no insulin. There's no point of having the receptor in the first place. So stimulate the production of the receptor is useless. The drug is scam. All right, let's go 42. The following best explains how small molecules move between adjacent cells. Now, in plant cells, they use plasmodesmata. So plasmodesmata is the commuting junction that is basically a protein hole that separates the two plant cells. There's a hole built into the cell wall, so things can 100% communicate, move between two plant cells. So the answer is B. So this is like, if you know what plasmodesmata is, super easy. Okay, animals use gap junctions. Plant equivalent is called plasmodesmata. Okay, it's plant's communicating junction, allows stuff to communicate. 43. Which of the following is likely to create genetic variation? We know genetic variation. So crossing over, independent assortment, right? Mutation. So which one is that? Will be D. DNA polymerase makes error, right? They do. So that will create genetic variation, yeah? We never talk about RNA polymerase error, okay? Because that will not change the DNA, all right? So easy one, 43 is C, DNA replication error by chance. Okay. All right, we have one more data in the set. Oh God. Okay, so it's like you expect to get quite a bit. Yeah, data in a set question. Again, those are usually the hardest. This is the last set. Let's finish this. Okay, so let's look at this one. To investigate the influence of predation risk on ray behavior. So predation, we talk about predation, okay? So this is ecology, predation, animal behavior. A student observed and counted large marine animals swimming in shallow, near shore section of the coral reefs. The shallow section, yeah? The time of each observation was recorded relative to the high tide. So when it's high tide, it's not shallow, right? The animal noted that low tide, when the water level is low, many of the large animals were forced out of this area into the deep water. That makes total sense. So when it's low tide, animals move to the deeper water. When it's high tide, animals move back. And our study area is the low tide. Right here we're told, shallow, near shore. Our study area is the low tide, not low tide, the shallow area. That is our study area. Okay, All right. During high tides, when water levels are high, larger animals are able to enter the study area. So your study area is the low, shallow water section near the shore. Okay. Over the 3-day period, so the student just sit there, put a camera there, and watch and count animals that came into the study area. So again, during low tide, very little animal, I guess. Yeah, high tide, more animals. So 604 individuals rays of 3 species were observed. There's a cow-tailed, giant, and black stingrays. So abbreviate them, guys. Cow-tail, giant, and black. So that's how I will be calling them. For each ray that was sighted, its body length was estimated. Okay, and the status as either alone. So alone means by itself, right? It's ungrouped, or found with others, with friends. Maybe they hang out together. Maybe they're alone. Okay. Occasionally, rays were observed sifting through the sandy substrate of the study area to capture food, such as mollusks and crustaceans. That's what they eat. So they dive their noses in the sandy area and try to find food. Okay. All right. In one instance, an injured ray with a bite mark that were likely sustained from a shark attack. So the predator sharks were sighted. In addition to the rays, students observed lemon sharks. So those are the predator, I guess, and also the black-tip shark. So there's 46 and 39 of them. So the sharks are there, presumably to hunt the rays. And maybe that's why the rays are grouped together, right? So they're not attacked by sharks. And body length, the bigger the rays, the bigger, the more likely they're not attacked, right? All right. So the result of the study are shown. So let's see what the result is. So we're giving all this background information to think about, and let's see if figure 1 makes sense. Yeah. Okay. So figure 1 says on the bottom, comparison of the body length of the grouped and ungrouped rays at 2 standard error of the mean. Okay. So I'm looking at ungrouped are longer. That makes sense to me because if you ungroup, you're alone, you're bigger. You're not as afraid. But if you small rays, you better be grouped together because the sharks are around. We have 2 types of sharks, right? The reef shark and the lemon shark. Okay, so this kind of goes with that. So the ungrouped one are longer and bigger than the group one. That makes sense totally for predators, right? Okay, so again, we're trying to study predation. Let's look at figure 2. The mean number of rays per group. So if they do group together, what is the mean number of rays? So do they group by 2s, by 3, by 4, by 5, by 6? High tide is T0, so this is high tide. So when there's high tide, why do you think they group together? That makes sense. High tide means that the water level is high, so the sharks are here. So you want to be grouped together to protect yourself. So that makes sense too. When it's low tide, you don't need to be grouped together. Why? Because low tide, there's no sharks. The water level is low. And group together actually help. You want to be alone to avoid competition. Group together provides benefit. What is the benefit? Benefit means that avoid predation, right? Avoid. The cost is only have so much food. Group together compete. About when there's low tide. When there's low tide, opposite is true, right? You want to be alone, so that you avoid competition, right? So that's what basically what this figure 1 and 2 is telling us. Oh, there's more? Oh, good Lord. There's figure 3 and 4. Okay. Now on the test, I would just kind of glance through these figures, okay, and then go to the question. But it is better if you get a sense of what they're trying to do. Like figure 1 is trying to talk about the body length, right? So if you ungroup, you better be bigger by yourself. And this is talking about the high tide. Yeah, if you're high tide, sharks are around, you want to be grouped together. Let's see what this one. This one is talking about the 3 types of rays. Remember the cow, tail, black, and giant? So it looks like high tide, there's more giant. Yeah. So high tide, the relative rate, there's more giant and black. So giant and black, there's more during high tide. Okay, for some reason. All right. I don't want to overanalyze this one. Let's move on. Maybe because they're bigger than cowtail. Okay. And then the last one here, I have sharks. All right. These are the sharks now. We have lemon shark and reef shark. Okay. So we have these sharks, and looks like high tide, there's a lot of black tip. Reef. Reef sharks are there hunting during high tide, and the lemon shark hunts during low tide. All right, so there's a difference in preference, right? The predator has different preference. All right, number 44. Which of the following claims about the survival strategy used by organisms in a coral reef system is supported by data in figure 1? So figure 1, 44, we talked about it. 44, the answer key says is A. Smaller of a vulnerable population are more likely to group, act collectively. That's a fancy way to say group. And again, the data shows that way. Because if you're on group, if you're grouped, you're smaller. See that? So you can have a herd advantage, protection, mutual protection. All right. So small one group, larger ones don't need to. So that's what figure 1 is supporting. 44 is A. Right? 45. Based on the data in figure 2, which of the following behavioral pattern do rays in coral reef typically exhibit 3 hours before high tide? So this is figure 2, okay? 3 hours before high tide, that'll be this. Because 0, it says, is 0 is high tide right here, okay? So their mean group size is 1, so they don't group. 3 hours before high tide, they're alone by themselves. It's during the high tide, they group together in 4s and 5s, okay? So 3 hours before, they don't like to group. That was my first impression. So if they don't like to group learning, no. Mate selection, no group. Yeah, social interaction, no group. So has to be C, solitary. So they have to be by themselves, and presumption here is that they're looking for food. That you don't want to have a competition with everybody, right? So they rather be alone. 45, 46. So which of the following scientific claim about interacting populations of giant nose ray and black-tipped sharks is best supported by results on figure? Oh, good Lord. So we have the shark, we have the giant, okay? Sharks eat the giant, okay? So if you look at the figure 3 and 4, we are looking at the shark right here. So the reef shark loves to hunt when what? When they are high tide, yeah? Reef sharks hunt during the high tide, okay? So keep that in mind, and let's look at the giant nose. Giant nose, where's the giant nose? Giant nose is right here. They're around in the high tide. Okay, so giant nose are around in the high tide. And reef sharks hunt in the high tide. Okay, they both shows up around high tide, okay? So we don't know why. So let's read you have to read all the choice. So the question is, what is the interaction between the giant and the reef shark supported by the evidence? So they both show up in the high tide, 46. Some parasitic species, forget about parasites, probably not. Invasives, you know. Migratory species, some prey form protected group. Yeah, here, then. This is the only one. So 46, the answer is D. The prey, which is the giant, they form protective group in the presence of the predator. Okay, so that is only one that has to do with prey and predator. Has nothing with the migration or parasitism or invasive species. It doesn't even make sense. So even you just guess, the answer has to be D. The whole experiment is about predation, right? Predator and prey. 47, which of the following justify the use of study area to investigate how one species influences the behavior of the other? A, black stingray were present in the study area in the presence and absence of reef sharks. Giant rays and cow rays spend longer period of time at rest while inside the study area. Now we don't have this information, how much time they spend resting, yeah? Natural barrier in the study area, we don't know whether, there's only low tide. Cow tail in the study area form groups in some stages of the tide cycle, but remain alone in other stages. I don't think we have that data either. So let's see, black ray present. Yeah, it's either the presence or the absence. That's all we're doing. So A is the most general answer. We don't know whether how long they remain alone. It's either they're alone or not. We don't have that information on everything else. So 47 should be A. Black were present in the study area, and it doesn't have to be black stingray. It can be anything. It's just the presence or absence. That's all we're doing. We're not looking at the natural barrier environment. We're not looking how long they're alone, how long they're grouped together. It's just either they are or they're not present. Okay. So that will be that one. And the last one here in the study group, based on the results of the study, which of the following is the most likely connection between the behavior and evolutionary fitness? So to me, I think you want to be grouped together when you're small to be fit, right? To survive. So 48, rays that search for food alone at low tide typically grow to larger size. I don't know that, yeah. Rays that join groups during rising tide are reproductively more successful. That is good. Remember, when there's high tide, you want to be in groups because that's when the predators are around. So they should be more successful than rays that don't, right? So there's about behavior, okay? And rays that swim far from the shore at high tide, we don't know how far they swim from the shore. We only have one study area, so no. Rays that roam across large distance, we don't have that information either. So the best answer is B, about grouping. Grouping together improve your fitness, yeah? All right, let's look at just one more here. So phenotypes are determined here by part when genes are expressed. So it illustrates gene X. So you can see the product of gene X, which is protein X, actually can turn on gene 1, gene 2, and gene 4. Crazy. Yeah. So gene X can control gene 1, 2, 4. All right. The question is, which of the following best explains how gene X control gene expression? So 49 is B, right? Protein X is responsible for acting transcription of the gene. Some, but not all the others, right? Yeah. It's not processing mRNA. The mRNA is right here. We're not processing them. Protein X is a member of cytoplasmic protein complex. Well, that doesn't make sense. Yep. So it's just that it turns on few genes, 1, 2, and 4, but not gene 3. So that's as simple as what the diagram is saying. All right. Now, the reason I didn't go over the, there's a few more here. Yeah. I'm too slow. These questions are towards the end, are no longer there. The fill-in ones, they're not there anymore. So we don't have to focus on those. So there's no more fill in your answers, okay? So on the new test, all right? So the rest is pretty easy. Yeah, stupid, I didn't finish this. Okay, so again, you have the answers on the, on the Google Classroom. So next week is free response. Please work on Google Docs or Word and submit them. And right now we have to be more critical. You have to start doing tests, okay? Before, week 1, 2, 3, 4, we're gonna do it together, right? We finished most of it. You can look at the video, but I need you to start doing the test. It's very important now you'll be able to put your thoughts into proper writing and be able to see what you wrote down and edit your own responses, okay? So we have to do that. So do that as much as you can, please. Yeah, okay. And I'll see you guys next time. Thank you guys for coming. Okay, bye guys. Bye, take care. Bye guys. Bye guys.